There's been a lot of excitement in the press recently about some of the developments in the use of artificial intelligence for a wide variety of different tasks and in ways that apply to a lot of the jobs that people are doing. So we thought it would be an interesting experiment to see whether some of these new AI systems could actually be of any benefit to people who are doing bioinformatics. So we've been taking a look at these. Specifically, we've been looking at the Bard AI from Google and the ChatGPT from OpenAI, which is largely Microsoft funded, and just seeing what we can do with them. It's probably worth sort of clarifying exactly what we mean, because AI covers a wide variety of different sort of techniques and applications, many of which are already kind of well understood and well used and regular tools in the field of bioinformatics. So within AI, you can have everything from sort of conventional machine learning techniques. A linear model is arguably an application of machine learning, decision trees, random forests, all of those kind of things that are very commonly found in papers. Going on a step from that, those models are okay when you're dealing with kind of a handful of independent variables, maybe sort of up to sort of 10 or so, but they don't really scale to very large and very complex data sets. So for those kinds of analyses, people are moving to neural nets and the sort of systems of deep learning. And these, again, they're numerical models. They're just much more complex numerical models, where instead of building a single sort of numerical relationship, you are building whole collections of these and having sort of layers of inputs to different numerical models, collapsing the outputs of those, feeding the outputs of those into another layer of transformations, collapsing the outputs, feeding those through, and having this sort of multi-layered process, where the big difference is that although you can get a great improvement in the efficiency and accuracy of the model, the complexity of what they're doing really sort of moves you to a level where you can't try and rationalize and understand exactly what the model is doing. With simpler models, you can kind of see the working behind it, but at these sort of neural nets and deep learning levels, you're putting a lot of faith into the model and you're not really rationalizing the outcome of it. These models have been put to a wide variety of uses. I guess the ones that hit prominence kind of very early on were sort of using this to do things like play chess, where IBM had their Deep Blue model, which went on to beat the sort of World Chess Championship and prove that computers really could be better than that than humans were. It's had scientific applications. Probably the most impactful application of this sort of technology was done by DeepMind and their AlphaFold system, which really solved one of the generational problems in bioinformatics, which is the prediction of three-dimensional protein structure from sequence. This is something that we have been sort of plugging away at for many, many years, and with sort of limited success. The AI-driven approach that DeepMind put together suddenly took this a huge step forward, where now we have really quite good ability to predict structure from sequence using one of these sort of deep learning models. Now, we're not going to talk about any of those today. What we are going to talk about is a specific application of these models, which is feeding them data so that they try and understand language. Okay, so the system that's garnered most of the attention has been the ChatGPT system, but there are others that are all other people that are also building these large language models. Now, language models themselves are not new. Okay, we've had large language models for some considerable time, and you are kind of going to be used to seeing these in sort of everyday use all the time. So the language models that had existed kind of prior to this had been sequential large language models. So these are models that were trained on a large corpus of data, and they were probabilistic models which could look at a set of words and predict what the next most likely word was. So it was kind of a frequency analysis to say, well, when I have this collection of words before, what am I most likely to put next? So you could build something up. And the application where you'll have seen this all over the place will be in something like a Google search, where you type in the start of a query, how heavy is an, and it will then go, well, if you've started a question with that, the most likely thing that you're going to ask for are going to be adults' brains, elephants, electrons, Olympic bars. Okay, those sorts of things are probabilistically the words that you're most likely to put after that. So it's sequential in that it operates in a linear fashion. Another sort of demonstration of this, which is dead easy to do, and if you haven't done it, it's an interesting little thing to try, is that these are built into your mobile phone. So when you're putting in text into one of your messaging applications, you can give it an initial prompt, which can be something as simple as hello. And at the top of your keyboard, it will suggest the words that you might want to come next. And again, it will be a sequential probabilistic model. So if you can just sort of keep taking the most likely word, you can get it to write you a new sentence based on the most likely thing that you've said before. So you can say hello, and it says, hello, my name is, okay, fair enough, my name is the best friend. Okay, so you can see that all of these words are kind of individually linking. My best friend and I, yeah, okay, that's it. My friend and I have a great, okay, so what are we going to do? I have a great day, okay, have a great day out, yes, with the boys. You can see in this sort of model how every word is relevant to the words that preceded it. So, have a great day out, but that doesn't mean that it stays in context with the entire previous sense of the sentence. And this is the limitation with these sequential models. They're very good at adding sort of individual information based on the preceding few words, but they're not trying to get a sense of the overall structure of your sentence or the meaning of what it is you're trying to put together. So their application for generation of new text is limited because of the way that they work. The new AI models that are now coming out, though, are approaching this problem in a slightly different way. So these large language models will take a sentence like this. So here's my sentence. I prefer the morning flight through Denver. And rather than just treating it as a linear series of words, firstly, it's going to assign roles to the word. So it's going to understand the function of the word within that sentence. So we can say, okay, well, this is a pronoun, this is a verb, this is a noun, this is a proper noun down here. And then it's going to parse this to try and make the connections between the words. Language is not a purely linear string of words. It has structure, and that structure is often sort of referring back or referring forward. So here, if we parse this sentence, we can find the kind of root of the sentence. So this is a sentence talking about a preference, okay? Who's expressing a preference? Well, I am expressing a preference. What am I expressing a preference about? About a flight, okay? What do I know about this flight? The flight is in the morning, okay? Great. What else do I know about the flight? It goes through somewhere. Where does it go through? It goes through Denver, okay? On a large scale, therefore, you can feed a huge collection of text into these models, and it will build up these connections between words, between concepts, and again, it's a probabilistic model, but in a much more sophisticated way. This then allows it to do two things. So firstly, it builds up this concept of the linkages between types of words and the structure that would define language. And secondly, if you give it a prompt, so you give it a small snippet of text with some connections within that, essentially your question, it can then use the sort of weightings and the linkages within your question to generate new, previously unseen text from the large model that it's built, which will then preserve the sort of structure and concepts. So it will generate new, natural-looking text guided by the prompt that you give it. And that's what these large language models are now producing. So we get something as simple as a prompt box where you can type in a piece of text, which can be almost anything, but is generally phrased in the form of a question. And it will generate output using the linkages in that prompt. We can put anything in here, but obviously we as bioinformaticians are interested in what might be relevant for our field of work, and people are starting to publish stuff now suggesting that this could be useful for sort of automating parts of the tasks that bioinformaticians undertake, or informing us or helping us sort of learn new things that it knows about and we don't. So that's what we wanted to explore. And in terms of what we actually tried to get these models to do, we thought of kind of three main tasks where these sort of large language models could potentially be useful. One is going to be in the writing of code. Okay, so yes, these models were initially sort of designed for parsing natural language, sort of prose, as it were, but you can feed them other types of text, such as code. And code is highly structured, it has sort of very strong linkages between it, so it fits really nicely into this sort of model. And many of these systems now can generate code for you based on a prompt that you express in natural language. So we can see if it can do that. We also spend quite a lot of time writing little pipelines and running analysis where we're not writing new code exactly, but we are linking together programs which perform specific tasks to achieve an end. So we're sort of generating these little pipelines of connections to transform data in specific ways. And again, if we had some help with that, that could potentially be useful. Finally, we could also use these for understanding biology. If you're a bioinformatician, often you're working on many different projects in different areas of biology, and you can't be an expert in all of these all the time. You often get new things thrown at you. So these sorts of models could be a sort of shortcut to helping to understand new areas of biology that you're trying to start a project in. So we're going to have a look at that. And we've been looking at two of these systems, the ChatGPT system from OpenAI and the Bard system from Google. The first thing that we should say is that when you go to either of these websites to go and look at these, the thing that is presented to you front and center the moment you start are a bunch of caveats, and they do it in slightly sort of different ways. ChatGPT sticks up little boxes like this that says, this may generate incorrect information, it may produce harmful instructions or biased content, and it doesn't know anything after 2021 because that's when the model was trained, so if it happened after that, it knows nothing about it. Bard says something similar, albeit in a much more sort of wordy way, saying, yeah, responses may be inaccurate, you've got to double check things that are in there. So they're wrapping any response that they give in here in a lot of caveats, and we will see why when we actually try and use some of these. So we need to bear in mind what these are. They are not sort of general intelligences, they are language models making linkages between concepts. Let's have a look at them in action then. Let's start with writing code. There have been a few reports of people taking exams for various types of qualification and plugging them into these large language model systems and finding that they actually do quite well. So if we take programming as an example, you can take the questions from an introductory level programming course and plug them into ChatGPT and see how well it does. And in general, it does pretty well. They often find that it does at least as well, if not better, than many of the actual students that are studying in this. So potentially this is quite a useful tool for generating code for us to save us time and sort of effort on this. Most of the tests that have been done on this have been of the form of questions in exams or things that are fairly introductory in their level of complexity. So we tried it. We run courses, we run R courses, we run Python courses. So I've taken some of the questions that we ask our students in our courses and fed them to these systems to see what they would do with it. So the first one here, write an R script which puts Simon into a variable and then calculates the number of characters it has in it. I think this is exercise 1 in our introductory R course, introducing some basic concepts. In this case, I asked Bard to do this, and it says, sure, here's some code. And it writes me some code. Not only does it write me some code, it puts some comments in it to say what each of the lines is doing, and it gives me a thing at the end to say what it's going to generate when it does it. So, create a variable called name and assign it the value Simon. Yep, there's Simon, correctly quoted. An assignment arrow into a variable called name. Calculate the number of characters in the variable name. It uses the length function to calculate the length of name, and it says the output is 5, which is the number of characters in Simon. Great, that's what I wanted. I can copy that code, chuck that into R, and I run it. Those of you that are R people in the audience, what number does this come up with? It comes up with a number 1, which is not what it said it was going to do. The length function, in many languages, in Python, in JavaScript, in Java, in Perl, calculates the length of a string, but not in R. In R, it calculates the length of the vector in which the string is contained, and this vector only has one string in it, so gives me the length of one. So it's very confidently and very nicely given me an answer which is wrong. Okay? And hence the caveats at the start. So first thing to note with this is that these things can generate incorrect information. Even when they're telling you what they're going to do, they're not actually running this code and checking the output. They will confidently tell you one thing and it might actually do another. We can try the same thing in ChatGPT. So same prompt in here. ChatGPT says, certainly, here's an example of an R script that does what you asked. It assigns Simon to a variable called myword. That's fine. Here, it then uses the nchar function of myword to get the number of characters and prints it, and that's fine. Okay, that's the correct function to use to calculate the number of characters in a string in R, and it gets the answer right. Okay, so equally confident, but in this case more justifiably. In general, in the tests that we've done, we found that ChatGPT was getting answers correct the first time more often. It's had a little bit more feedback. It's slightly less new. Other systems will catch up as they get more feedback and more development. So, next question on here. Use the rseek function to create a vector of 100 numbers starting at 2 and increasing by 3 each time. We're probably about 3 exercises into our R course at this point. Again, gave this to Bard. It says sure. I don't think we've given it any programming task where it's gone, no, don't know how to do that, or I'm not sure, or can I answer these questions? It just goes, yes, here's a thing that you've asked for. And here's its code. Use the seek function, yes, and I want a vector of 100 numbers starting at 2 and increasing by 3 each time. And it gives me this version of the seek function, says the output is a vector of 100 numbers starting at 2, increasing by 3 each time. And if I run that, it's correct. It gives me the correct answer. That is indeed a vector of 100 numbers. It starts at 2 and it increases by 3 each time. However, it's done it in a slightly weird way. The aim of this exercise was for people to understand the arguments to the seek function, and I gave 3 very specific numbers. I want a vector of 100 numbers starting at 2 and increasing by 3 each time. So really I wanted a version of this function that used the numbers 100, 2 and 3, and that's not what it's done. This has actually done the calculation internally and said, well, if you start at 2 and you go up by 3 each time, then if you go to the number 300, then you'll have ended up with 100 numbers in your vector, which is correct, but it's not what we wanted people to get from that exercise. We wanted them to find the argument that says, I want the output to be 100 numbers long. So it's correct, it generates the right output, but it's slightly missed the point of the question. Again, trying this in ChatGPT, ChatGPT gets the answer that we would want the students to get. It uses from equals 2, by equals 3, and then length.out equals 100, which would be the way to constrain the length of the output. Identical output to the previous version, but more correctly getting the sense of what we were trying to get the students to do. So, second takeaway from this is that you can get code that might answer the question, but it might do it in an unusual or slightly weird way. Third programming prompt we gave it was this. It says, create a data structure to hold the following genomic positions taken from the human genome. We gave it a bunch of positions, chromosome, start and end. And then we said, generate random genomic positions in a loop and check to see if they fall in any of the regions listed above. Okay. Sure, can do, says Bard. And it makes this. Now, this is a correct answer to the first part of this. Here is a Python structure. We've moved it to Python here rather than R. Here is a Python data structure that holds those genomic positions, and it does. It chooses to make a dictionary out of this, where it uses the chromosome name as a key, and it uses a tuple of the start and end values to hold the position on that chromosome. So technically, it has done what I asked it. But conceptually, it's not done it in a very sensible way. So the set of genomic positions I gave it happened to all be on different chromosomes. But I certainly didn't say that these sets of genomic positions will all be on different chromosomes. So it certainly works for the input data I gave it. But if I now take this concept and say, right, here's another set of positions. Now there are like 3 positions on chromosome 1. If I replicated this with chromosome positions on the same chromosome, I'm going to get them overwritten because I can only have 1 position on each chromosome with this structure. So technically correct, but it hasn't understood the concept of why I might want to hold genomic positions and what to do with it. Incidentally, I fed this one into ChatGPT. ChatGPT did exactly the same thing. It also made a data structure with chromosome as key and start and end positions as value, and missed the point that you might have multiple positions on the same chromosome. So kind of, yeah, technically correct, but not really understanding the concepts behind. Then for the second part, we'd asked it to generate random genomic positions. Now, I told it this came out of the human genome, so it would have had to have done a bit of inference to figure out that I needed to select from that. And Bard makes a complete hash of this. It writes me a function saying generate random genomic position, but what it actually does is it selects a random chromosome from the data structure that it built, and then picks a random position from between the start and end positions. So you can imagine it having seen many bits of code that do something like that, because often that's what you might want to do with this sort of data, but that wasn't what I asked. So here it makes no sense with what I said, so I'd asked it to check whether that region falls within the positions that I'd specified, and it's always going to, by definition here, okay, because it's picking one of the chromosomes I have and picking a position between the start and end. So not really doing what I asked at all. Okay, so again, misses the point, gets the answer wrong, generates me code that works, but it doesn't do what I asked it to do. For ChatGPT, I went a little bit more specific then and said, right, generate a random position within the GRCH38 human genome assembly. So write me a function to do that. Okay, and one of the other takeaways from this is that the more specific you are about the prompt that you generate, the more information you give it, the more likely it is to get this right. And ChatGPT has a pretty good stab at this. So first thing that it does is it makes up a dictionary of chromosomes. So these have the chromosome name as the key and the length of the chromosome as a value. I didn't give it these numbers. It's found those within its knowledge base, and these are the correct lengths for the chromosomes in the GRCH38 human genome assembly. Okay, so well done it. It's found that that's a relevant thing. Then to generate a random position, it selects a random chromosome from here, from the keys out of the chromosomes dictionary, and then generates a position between the first base and the length of the chromosome, and then it generates an end position. I didn't ask it to generate an end position. I said give me a position, and it's given me an extent, but at least it has done the sort of main thing that I asked. So on the face of it, yeah, it's done what I asked. However, selecting random positions in the genome can be quite a sort of complex task. If I generated a whole load of random positions on here, let's say I generate 1 million positions with this, and I look how they are distributed across the genome, I will not find positions equally distributed across the genome. The method that it is using to select these positions will introduce a bias. Its method is that it selects a random chromosome first and then selects a random position within that chromosome. And the chromosomes within the genome are not all the same length. Some of them are much, much larger than others. So because each chromosome has an equal likelihood of being selected, the positions within small chromosomes are going to be overrepresented compared to those within large chromosomes. Now, that's a logic error in here, or a logic limitation in here. This is still arguably a random position. It's just not completely random in terms of all positions in the genome being equally likely. So spotting this is not trivial. You have to understand exactly what it's done. You can't just take this code and run it and be able to spot from some kind of error message that it's not doing what you want. It's quite a small and subtle thing, but if you're running a big simulation, it's going to introduce a bias into your simulation. So you are not absolved of responsibility of understanding the method that it's using because it might have an effect on your data. One of the nice things about these systems is that you can kind of iterate on your immediate prompt. So you don't have to ask it a new question. You can say, oh, let me refine the question that I gave you before. So here, having seen this code and figured out that, oh no, that's just going to pick positions on smaller chromosomes more often, I can tell it that. I can say that will select positions on smaller chromosomes more often. Can you make each genomic position equally likely? And it goes, oh, yeah, here's a new version. And the new version does that. Okay, so now instead of picking the chromosome first and then picking the position, it adds up all of the chromosome lengths, gets the cumulative length, picks a position within that, figures out which chromosome that position would fall onto and which position it is, and then would return that for me. Okay, so with another prompt, it does better, but I had to spot the limitation in its code. Even now, this is not necessarily a perfect solution, so it is selecting from across the set of chromosomes, but I have an X and a Y chromosome in here. Now, if I'm selecting from a normal diploid genome, I have 2 copies of all of my autosomes, but I'm only going to have one X and one Y. So this would select equally if I was talking about a haploid genome, but not a diploid genome. Also, I didn't tell it I wanted to select from a male genome. I might want to select from a female genome, and that's not in there. I might want to include the mitochondrial sequence, and then how many sort of copies of the mitochondrion do I include in this? So there are other levels of complexity that this is not able to identify, and it's up to you to be as specific as possible in framing the question and indeed testing the output to ensure that it's doing exactly what it wants. And simple tests are not going to cut it for this. If I run this, I will get a position, and if I run it again, I will get a different position, so it looks like it's generating random positions. It's only through an understanding of it or a very large-scale simulation that I could spot the biases. People have realized these limitations in the models, and this is a diagram from the report underneath, where someone has suggested a sort of workflow for getting these large language models to write code for you. So the idea is that you come up with a prompt, a question to ask it, get the model to generate you some code, you run it and see if it works and does what it's supposed to. So that might just not run at all. It's possible that it will give you code that crashes or comes up with an error or doesn't produce any output, something like that. Or if it does produce output, you can evaluate that output and see if it matches what you expect. And if you get errors or you get an unexpected result, you can go around, iterate your question, refine its prompts, tell it what it did wrong, and get it to go around again until you get what you expect, and then you document it and use it. And on the face of it, that seems like a pretty sensible way to go, and actually fits with the way that you would develop code if you were doing it yourself. The issue with this is going to be that there are 2 steps in here that are quite hard. So if you have errors in this, you need to know enough about your language to understand what that error is telling you so that you can understand why it might have failed and therefore how to craft a prompt to do it. And just understanding that logic for what it's trying to do and why something might have failed when you haven't written that in the first place, you're reading its code, can be quite a time-consuming task. Okay, so you're not absolved of the responsibility from this, and actually reviewing somebody else's code is often much slower than writing it yourself, because at least when you're writing it you have your own mental model of what's there, and reviewing someone else's requires you to build that up as you go through, so it's quite mentally taxing. Also, from the expected result, it depends how obvious it is that that result is correct. Okay, in some cases and some options, it's quite trivial. Okay, yeah, you know exactly what you should get. Yes, I got it. It's all good. Sometimes it's not, like in the case of the random genomic positions. Yes, it generates random genomic positions, but they're not quite done in the way that we would want, and there might be some sort of logical error. we all know when we're writing code, things like it's easy to have sort of edge cases. What if I select the last base on a chromosome as my random position? Oh, well, then my end position might not work. Have I handled, when I'm reading in a file, have I handled the last line in the file correctly? And it might be that all the rest of them are fine and it misses off the last one. So validating that fully can also be a non-trivial task. So the trade-off here is whether going through this sort of process overall saves you time over how long it would take you to actually run this yourself. And yeah, we'll see how that plays out over time. We've also tried running these models for running analyses, and actually what I did was just over the course of a couple of weeks, I just took any job that I was doing and said, right, what if I'd asked one of these language models to do that for me? What would it have done? Would it have done the same thing I did? Would it come up with the right answer immediately? Would it have saved me some time if I asked it to do that? And I've shown plenty of examples of places where it didn't work, so I'm going to show you an example where it worked really very nicely. One task that I had to do was to take an unsorted BAM file and turn it into a bigwig file so that you can view it in sort of genome browser systems. And there are potentially lots of different sort of tools that you can use to do this and do different steps. And if you've never done it before, there's going to be a fair bit of Googling to figure out what you connect together to make that happen. ChatGPT, when asked about this, came up with a process which was perfectly sensible using tools which were all there, and gave me an example code which did exactly what I wanted. So it said, yeah, you need to sort your BAM file using SAMTool, so it's sorted by genomic position, convert that into a bed graph format, so it used bed tools to do that, and then use bedgraph to bigwig to convert that into bigwig format. Okay, so yeah, here's a command to do the sorting. Here's a command to do the genome coverage on the sorted BAM file, generating the bedgraph file. And here's the command to take the bedgraph file and turn it into my bigwig file, which is what I wanted. So great. That did the right thing. And if I run that, that would work. There's a couple of little sort of things that were not enormously clear. So things like in here, it says, I need the chromosome sizes, and it doesn't tell me where that file can come from or where I get that information. In fact, those chromosome sizes are going to be embedded in the header of my BAM file, so I can extract them from there. So it doesn't say that. So I'm still going to do a little bit of additional work in that I'll need to find where that is. But generally, that's been pretty good. So here is a good example of where it would have helped me and saved me time. This was another project I was working on. I spent quite a lot of time on this. This is probably sort of half a day's worth of playing around and doing stuff. What I'm looking at in here are two single-cell RNA-seq data sets, and I was using some of the functions in the SORAT package to integrate them together, so to do kind of batch correction and align the data from the two data sets to make them more comparable. Part of the process for doing that is that you generate what are called integration anchors. So an integration anchor is a linkage between a cell in one data set and a cell in the other data set to say these two cells are of kind of equivalent type. Okay, and you do that across your whole data sets. You make up a whole set of integration anchors, and those are what are used to then sort of numerically skew the data to make them line up. And I wanted to be able to see the integration anchors, so you can calculate these fairly straightforwardly and run them through the pipeline, but it's all a bit of a black box, and I wanted to see what it was doing. So I'd spent quite a while working out some code that would then let me look at the integration anchors between the red and the blue data sets, so I can say, oh yeah, look, these are all lining up with things here, these are going over there, these are mostly sort of within that cluster, these are sort of linking together here, and it was quite a nice little visualization. So I thought, well, that was quite a complex thing for me to do. So how would the models do on this? So I said, how can I visualize the integration anchors when integrating single-cell RNA-seq datasets in SORAT? Okay, great. It says, sure, here you go. What you do is you load the SORAT that contains the integrated data, use the find integration anchors function to identify the integration anchors. Well, yeah, that's what I'd used, so that's what it did. And then I used the plot integration anchors function to visualize the integrated anchors. And I felt like a complete fool. How would I not noticed that there is a function already in SORAT to plot the integration anchors. Okay, not only that, gives me some data on here, some code that says, yeah, you find them, then you plot them. Not only that, it tells me what the function does. It says it looks at the distribution of anchors, gives me information from the sort of help from plot integration anchors to say exactly what it does and how to interpret the results from it. I'd wasted half a day. Okay, I've done all of this. I could have just run that. So went off just to look up the documentation for this. So I Googled for Surat and plot integration anchors and found nothing. There is no function called plot integration anchors in the Surat package. As far as I can tell, there's no function called plot integration anchors anywhere. It doesn't exist. The program had tried to give me an answer to my question, and it couldn't find an answer that was what I wanted. So instead, it makes stuff up. And it makes it up based on its large language model, so it writes things that look very convincing. So I get examples of code, I get nicely written text, I get snippets from documentation, and all of it looks convincing, but is not real. I tried the same thing in ChatGPT. ChatGPT said, no, not plot integration anchors. What you need is the anchor plot function. Okay, maybe Bard had just made a mistake, linked to the wrong function name. It had done that before. Find integration anchors and then do the anchor plot with the integrated data. And it tells me again what it does and how this is going to work. So, okay, well, maybe that's it. So I Google for that. That also doesn't exist. So these models all suffer from the same thing. And it's something that the people that build these models know about. They are not trying to give you accurate information. They're trying to give you sort of convincing information. So they tell you in the warnings that these systems can hallucinate and present inaccurate information as factual. They have invented names of functions and bits of documentation and code because it sounds like that should be the answer to your question, even though those functions don't actually exist. And this is a known and really problematic side to these, to the extent that some of the main sources of information that we rely on for sort of technical answers to things are becoming corrupted. Stack Overflow, like the biggest collection of sort of technological programmatic answers to questions, has had to ban people from submitting AI-generated answers because they are often incorrect. And once it gets fed into that sort of corpus of information, it dilutes the value of the data that we have there. The issue that we have is that a lot of the way that we judge the likely validity of a piece of information is how well written it is. So if you get something that's written in sort of poor English with very few examples and it's just a little snippet, you go, oh, okay, well, it could be right, but it doesn't sound great. If you get something that's nicely, beautifully written and set out and nicely punctuated, and it's got kind of references, and here's a snippet of code and here's a bit of documentation, we inherently are likely to believe that because it looks like someone who knew what they were doing was writing that out. But large language models are extremely good at formatting it nicely, presenting it nicely, but can be mistaken in the actual information that they put in. So one of the things we're going to need to do with this is to kind of reset our brains for how much we trust answers and think about other metrics that we would want to employ other than how well written they are. Finally, I wanted to look at some biological questions. I often get faced with new pieces of biology that I need to understand, and going around different textbooks or websites or papers to summarize a whole load of stuff to get a feel for a new area of biology is quite a lot of work. So that's again an area where these models could be useful. As it happened, I was doing a job and the group leader in question had given me some information about a particular area of biology, and I thought, well, I'm going to use these models to see what they can tell me about it. One of the things I'd been given was a list of genes that were relevant to this area of biology. So I just said, well, tell me what these genes have in common. Okay, I knew what they were supposed to have in common because the group leader had given it to me, but I hadn't told the model. So put those in, and it said, yeah, these genes all code for proteins involved in the formation and maintenance of connective tissue. makes up cells, fibers, and extracellular matrix, and it's a network of proteins to hold cells and fibers together. And that's what these things were. These were genes linked to the extracellular matrix. Now, it's found that linkage. It's given me a summary of what these different gene categories do, which is great. There's a real shortcut. Not only that, once I knew we were talking about the extracellular matrix, I could ask it questions about that. So, well, what is the function of it? And it gives me really nicely written, bulleted summaries of large amounts of information that tell me an awful lot. It tells me that this is an attachment surface. It can signal into cells affecting behavior. It provides structural support, involved in wound healing, also involved in diseases and other stuff. Great, all useful stuff that I can do. I can even go in more depth. How does it signal to cells? Yep, great, tells me about sort of cell surface proteins and how those come in through soluble secreted factors and growth factors. And even via sort of mechanical forces. So again, all really good stuff. And if I want to go one level deeper, which genes are involved, and I know that many of the things that come up here are right because they link back to what I've been told before. So yeah, integrins, growth factor receptors, transcription factors, tyrosine kinases. Brilliant. Really quick, really accessible way to get in. Now, I didn't know much, I still don't know much about the extracellular matrix, so taking this stuff at face value is great, and I think I have learned quite a lot fairly quickly with this. But I didn't know how much I should trust it. If anybody who knows a lot about extracellular matrix cares to pick apart any of the things that are in here, I can add those to the comments on here. So I decided to ask it about something that I did know about. Okay. We've done a lot of work on epigenetics and DNA methylation, and one of the questions that comes up a lot is kind of how pervasive a mechanism is the use of DNA methylation to regulate transcription. Okay. I know that many people have ideas about this, and that answers for how prevalent this is can vary, so I'd be interested to know what the system made of it. Bard kind of dodged the question. So it said, oh yeah, it's a major mechanism, what it's involved with, how it works, various things in there. And then at the end it says the extent to which DNA methylation is still being investigated. Okay, so it's kind of skirted it. But again, at the face of it, a good sort of technical summary of what we've got. However, when you look in a little more detail, it's not all great. So when it was looking at the description of this, it's the addition of a methyl group, CH3, to a cytosine base in DNA. Yes, it is. This can occur at the 5 position of the cytosine. So yeah, 5-methyl cytosine is the one that happens. Or at the 6' position. And that was news to me. I'd never heard of 6-methyl cytosine. So again, I'm thinking, have I missed something here? Is there a variation on this? And it even says, like, methylation at the 5' position is more common and thought to be more stable. So maybe this is something that I'm learning from doing this. That there is 6-methyl cytosine that's a bit more rare and maybe, a little bit more niche, but now I've got something from it. I've gone and have a look, and as far as I can tell, 6-methyl cytosine is not a thing. It's certainly not a biologically relevant molecule in any way. 6-methyl guanine or 6-methyl adenine, yep, those are things, they do stuff. 6-methyl cytosine, not a thing. So again, within a corpus of text that generally is accurate and nicely summarized, it throws in these little nuggets of stuff that it's just made up. Probably aggregating from other related information that doesn't really transfer onto here. So the devil is in the details with this stuff. Spotting the things that don't fit in something that I knew a lot about, yeah, I can do that. In the other corpus of text, not a chance. I wouldn't know which was the little sort of fact that got thrown in that was made up. I decided to be a bit more specific and say, right, give me some numbers. How many genes are directly regulated by DNA methylation? And it goes, yeah, sure, I can tell you. It is estimated that 80% of all genes in the genome are regulated by DNA methylation, and that it plays a major role in determining which genes are turned on and off. That seems like a big old number. It's a very definitive number, and it seems very confident in that. But we know that we have cell lines where we have deleted the methyl transferases. So essentially you remove all methylation from the genome. And some genes go absolutely nuts because their regulation gets messed up, but most genes couldn't care less. They are perfectly happy with or without DNA methylation, and certainly not 80% of all genes in the genome get affected by that. So that seems quite a contentious number. One of the things that you have in here, though, is that in the top, there's a little thing that says view other drafts. So I thought, well, let's have a look at the other drafts it had of the versions of this answer. And the second version of this said it's estimated that 60% of all human genes are directly regulated by DNA methylation. So just by going to the next answer down, I can already get a change in 20% of my estimate. So maybe it's not a number that's very sort of strongly backed up, or maybe there are multiple sources that are claiming different things. I decided I would try and sort of query it some more so I can go back and iterate on this. So I asked it. I said, well, look, you've told me that 80% of all genes are regulated by methylation. Where did you get the figure of 80% from? And to its credit, it told me. So, yeah, it comes from this study, a study published in Nature in 2012. Pretty good source if you're going to quote something. So if it can back this up, that's all good. Tells me who did the study, even gives me a reference for this. Bernstein, Michelson and Zhu, the Human Epigenome Project 2012 update. Sounds like the sort of thing that would have that kind of information in it. Gives me the citation in Nature. Okay, please let me know if you've got anything else. What more could I ask for? Well, went off to PubMed, stuck in Nature 489-7414, pages 179 to 181, and it's not this paper. That paper, as far as I can tell, does not exist. There isn't a paper called the Human Epigenome Project 2012 update. These are real people, Bernstein, Michelson and Zhu. They are all people who publish around the field of epigenetics. They're all relevant to this sort of data. Bernstein and Michelson have certainly published together. I couldn't find anything where the three of them had published together, certainly not anything around this sort of time series. So it has aggregated together related and believable information to make something up, to come up with this 80% number. Doing a little bit of digging, I think I found where it actually got that number from. So if you go and look in Nature in 2012, there is a paper which involves Bernstein and Michelson and lots of other people, and neither of them are the first authors on it. And it's this one, which is the main ENCODE project consortium update. Okay, Encyclopedia of DNA Elements in the Human Genome. Bernstein and Michelson are both on that. Zhu, I don't think, is on this one, but there are tons of authors in it. And within that, it has the magic 80% figure. But it says this data enabled us to assign biochemical functions for 80% of the genome. It certainly didn't say that 80% of the genome was regulated by DNA methylation. So it is making linkages and connections, but in this case, it doesn't have enough of those connections to correctly generate text on the basis of this, and it mixes together concepts and gives me answers that are not right. So, again, if I'd read the warnings more carefully, it will tell me that it can make stuff up, but more than that, it will say that it misrepresents how it works. So I asked it where it got it from, and it told me something, and it lied. In response to asking how it was trained or how it cited sources or gave it fresh information, it just makes stuff up at that stage. It can't explain its own working. And it's not just questions from me that have been misrepresented by this. Recently in the news, there was a case of a New York lawyer who was preparing the legal work for a case that he was doing and had used ChatGPT to ask for legal precedents that were relevant to the case that he was putting together. He got a whole load of cases. And when he presented this to the court did some checking on it and said, well, look, you've cited all of these legal precedents and we can't find these. Can you tell us where you got it from? And it turns out that ChatGPT had just made this stuff up. And in his kind of defense for doing this, they showed the chat logs that he'd recorded for doing it. And he'd actually gone back to the model and said, oh, that looks like it's a really relevant thing. That would be great. Is this a real case? This isn't something you've made up? And the chatbot said, yeah, it's real. It wasn't. And he'd asked where it came from, and he, oh yeah, you go look in these legal databases and it's all there. And he even asked it, said, well, do you make stuff up? Are you making fake cases up? And it said, no, I'm not. And it was. So again, these models are not about presenting accurate information. They're about presenting nice looking prose from the information sources that it has. So what's the conclusions from all of this? Okay. These models are really good at giving quick and readable answers to questions that summarize huge amounts of data. So if you are trying to get into a new field, they will give you a very readable, compact summary very easily to give you that information and can summarize across wide ranges of sources. And where you're asking about something with a lot of kind of supporting information, where those connections have been well built up, those answers are likely to be very good and a simple way to get into that. The problem is that where you are asking a question that is not well known and where it hasn't had a lot of sources that specifically relate to the question that you have asked, it will rely on other connections between phrases in your prompts and will make up linkages to generate the text and will produce very plausible looking answers that are not actually grounded in reality. These things are designed to make nice looking text. They don't really understand the sense of your question, and they're not trying to parse anything beyond exactly what you've given them. So one lesson is certainly don't let it assume anything. Be as specific as possible in the prompts that you provide to give it the least possible wiggle room to make connections outside of the area that you're asking about. Whatever you get out of them, don't believe it on face value. If you're going to act on this, or you're going to sort of take any sort of steps and do work on the back of it, always go hunting for sort of secondary sources of information to confirm that the claims that it's making actually stack up. Okay. This can be time consuming. It's time consuming debugging code and validating it. It's time consuming rechecking facts in text that's come out. And the balance that we need to strike is whether this is going to save you time over doing that without the aid of the large language models in the first place. These models are definitely getting better. I mean, even between the Bard and the ChatGPT, we can already see a development where ChatGPT gives you the right answer more of the time in the first prompt than Bard does. Bard will catch up and these models will improve over time. The problem then will be that although they will be right more of the time, they're still going to throw in that curveball where they're going to get something completely wacky, and that we are going to become less sensitized to that. As we get used to them being right, we're going to find that the occasional time that they throw that in, we might miss those and we might get misled by it. So how this plays out in future, I think is still kind of open for debate. We will see if they can get to the point where the correctness of their answers is good enough that it actually is saving us time overall, or whether the inaccuracies in the end catch us out enough that we're better off referring to more reliable primary sources and not relying on these sorts of models.